Good morning. How are you? Need I ask?
I had my first dose of the COVID shot, so my arm is like so hurt.
Sore? Oh, okay. Is this your very first one? It's not like a booster or anything?
No, very hurt.
Okay.
I have this thing where I feel like I'm the ratio of 1 in 5. So I feel like all of that stuff happens to me. So every time we used to put the flu shot, I would land like the whole week in bed.
Oh,
So for me, like, to do the whole thing, I was just so terrified and scared. I'm not scared to die, but I'm just scared to get sick. I don't know how that works, but it's just weird. And my daughter, it's a requirement for high school. So
What high school?
she wants to go to the high school, so she said she would take it. It's the rate. It's over at
Oh.
19 and national.
national, yeah.
Yeah,
They
they,
require it?
because they do a workshop, so they can start going to work as soon as they're freshman year. And it's a private Catholic school, so that's what she's wanting to go to that school for the longest.
Mhm.
She got admitted, and then when they sent us the email that she needed the vaccination or whatever for work, I guess. I told her, "Are you sure you want to go to high school?" She said, yes, and she took it, and then my other son is like, well, if she's doing it, I'll do it too, and then once I was there, I was like, well, they started telling me, Mom, you're so dramatic, just take it, it's not gonna kill you, it's supposed to save you, and I'm like, I just went with the flow. I would- I would
Never got
think
the flu
that-
shot.
Yeah, it's been 8 years since I've taken the flu shot, so my fear was like,
Copy was on, and I wanted to be told. And I've not gotten it, I still was pretty sick. I have quite a few conditions. So my doctor was like, had I not had it, I probably would. But I'm still pretty sick. But I have a lot of.
Yeah, hospitalized after getting the flu, so I think it's supposed to be.
that's
Well,
what I was
yeah,
going for.
It may be some of the numbers, I say. But I have, I work with prayer. Also, my daughter works in Luke's. And the numbers of the ones that aren't vaccinated.
And mine was the opposite.
Yeah.
People tell me that I think I got vaccinated, ended up in the hospital, and the people that didn't get vaccinated, we were like perfectly fine. I'm just doing it because my kids took it, and they're like, well, you're gonna let us go and do this by myself, and I was like, I'm supposed to be a parent and be the role model, so I took on for the team.
All right, teachers on, my youngest son, he didn't.
Oh, you didn't?
No. he didn't. Okay. Well, I didn't see anyone online. What's going on? Let me see if maybe somebody has logged in now. Okay. I got one there. Well, we're down to the wire, right? Last two chapters, chapters 23 and 24. Now, when you read through the chapters, you will notice that it's like stuff that we've gone over. It's just a reiteration, and it's putting it all together for us. And in chapter 23 here, we'll discuss the body and the continued metabolism that's going on in the body. After death, we'll explore the metabolic reactions. And this chapter here talks about those metabolic reactions that it can cause the body to change. And also we'll look a little bit about, which we talked about before, the postmortem, the pH shift, from the neutral to acidic back to basic. We'll take a look at that. We'll look at the role of ATP in rigor mortis, which these are things we've looked at before. So we'll also take a look at the chronological journey or the order in which decomposition, the tissues in the body in which they decompose. So we'll take a look at that. So key concepts. So in death, entropy takes over, rigor mortis, gut bacteria, translocation, loss of youthful good looks. The chemistry of decomposition: carbohydrates, proteins, lipids, keratinized tissues, bones. So this, which we'll talk a little bit about, but this is kind of the chronological order in which the tissues begin to decompose in our bodies. Last unit, last two chapters, which we're going to be doing today. This is chapter 23. We'll be doing 23 and 24. Last two chapters in the text. Death. So, life is the act of achieving entropy. Because we said, like in death, decomposition, that's what it is, entropy. Entropy we know as being chaos, right? Our bodies, they consume other life forms as a source of fuel and use metabolic reactions to create energy by combining this fuel with oxygen. Remember we talked about, when we looked at the stages, we talked about a mechanical, chemical digestion, and then what happens when the bigger molecule is broken down. And so, and then how we, at the end, we extract and get our energy. Okay? And so that is what we do. And during that process of creating energy, we see the combining of this fuel with oxygen that we saw in that process. Oxygen is made in the process of ATP, which is where we extract the energy from in the electron transport chain. So the unwinnable battle against entropy comes to its inevitable end upon the arrival of death. Okay? So upon death, all energy in our bodies disperses like water bursting through a broken dam. All death, regardless of its origin, is ultimately caused by a lack of oxygen to the brain. Okay. So when the heart no longer pumps blood, the blood succumbs to gravity and flows to the dependent portions of the body. And that we know as the term livor mortis or lividity. Okay? And so that's the blood that pools settles to the dependent parts of the body. So with the heart no longer pumping, the cells no longer receive blood and oxygen. And so blood, which again is no longer being pumped, it succumbs to the physical properties of gravity. And so that's in that process, onset of death. Then we get lividity, and that's livor mortis, the settling of the blood to the dependent parts of the body. Continued metabolism. In the absence of oxygen creates lactic acid, which forms from pyruvate. And so you remember when we had glycolysis, and then we had the pyruvate conversion, then from there to the Krebs cycle and so forth. And so there were two examples of the pyruvate in aerobic conditions or anaerobic conditions in the presence, so that's the presence of oxygen, or without the presence of oxygen. And so in the absence of oxygen, that's when that pyruvate goes into lactic acid storage, right? And the other one, in the presence of oxygen, remember, it attaches itself to the acetyl coenzyme A, which goes into the Krebs cycle. So as proteins, as they break down, nitrogen compounds are created, which are themselves basic, and which also further break down into ammonia. You remember in the process, we talked about even with postmortem caloricity. And remember that was due to continued metabolism. And then when we looked at the pH changes, you remember it was neutral. And due to continued metabolism, it took the body into an acidic condition. And at the onset of decomposition, where you're having that breakdown, okay, of the proteins, and it was producing those nitrogens, the ammonia, and so which takes the body back from that acidic condition back to basic. Okay, and we've gone through that. You know, we went through that, and a lot of this is just a reiteration of what we've gone through. Rigor mortis sets in as well as the lack of ATP prevents the myosin head in the muscle fiber from releasing the muscle filament. And we went over that in chapter 19. And as decomposition further progresses, it breaks up rigor mortis. Okay. So eventually all the cells in the body die, and this is when the real action begins. All the cells in the body die eventually. So bacteria, gut bacteria, okay? So the infestation begins in the cecum. And this picture here is showing you know, the cecum, the area that's highlighted there. And so that's when infestation begins. And so they use the liquids that have leaked of damaged cells as a food source, okay? And so over a period of time, gut bacteria digest the entire intestines and the surrounding tissues from the inside out, and that is called translocation, okay, that process. Bacteria eventually starts to invade the capillaries of the digestive system, the lymph nodes, the liver, and the spleen, and finally the heart and the brain. It is during that period the individual doesn't look their best. So in short, there's thriving, aggressive, thriving life after death. Okay, it's the life of the bacteria, it's not the life of the deceivers. unfortunately, right? So now, the chemistry of decomposition. Decomposition depends on a lot of factors, a number of factors, including temperature and moisture. Remember, we talked about those factors, right? We talked about temperature, we talked about moisture, we talked about extrinsic, intrinsic factors, we talked about trauma, pathology. So decomposition depends on a number of those factors. Now, we do understand that embalming fluid delays decomposition. But what on a molecular level is it delaying? Okay? So we'll look at that, and that is what the remainder of this chapter will be looking at. Now, our embalming fluids that we use, they have two main purposes. Okay? So when we talked about, you remember we talked about functional groups? Remember those functional groups, right? And the aldehyde serves as a disinfectant and a preservative, right? And our main purpose for embalming is to disinfect, preserve, and to sanitize, right? Restore. Okay, so disinfectant, it does that, which is to kill the bacteria that both causes and contributes to decomposition. So that's one of the things that our formaldehyde and aldehyde does. Okay, and to hold together those biomolecules that have already began to degrade. Okay, so that's the dual purpose, the two main purposes of our formaldehyde, that aldehyde functional group, and that's to kill the bacteria and to hold together the biomolecules that have already begun to degrade, to break down. We'll take a look of the chemistry of embalming in the final chapter, chapter 24. In this chapter, we take up the chemistry of decomposition. Okay, so the decomposition, well, good morning.
There's an accident right under the 68.
No, the
No, there was like some, there was like
Yeah,
stuff,
nice
going, it was
BMW.
right before the exits get off here, and I was like, oh, you gotta be kidding,
You're
and I
the
realized how
lieutenant
close I
waiting.
was. So I was
Oh,
like, thank God,
me?
because it
Yes,
was
it's
stopped
been a
from
while
all the way
since
back, but I was in the farthest right lane, so I could
Okay,
just
so
go.
you can just go
I
ahead.
passed about a mile of stopped traffic, and then I could see how far it went forward. I was like, oh my gosh, I think I was in Milwaukee today.
I know when we were talking about it this morning, me and one of the other instructors, and he was like, Julian, it's just like people are driving crazy. He said, I said, yeah, they should be used to rain.
Yeah.
And you know, the weather conditions we have here in Wisconsin, they should be used to that. But people were driving crazy.
Zipping in and out, I'm just,
Yeah,
I can get anywhere
that
faster.
worries me with that. People drive so crazy. And they do the whipping back and forth.
Yeah, the Kia, you see that in Milwaukee, the Kia
Always in
boys
a
are.
hurry.
Yes. It's just like, and sometimes I just like, stop.
Mhm.
Y'all do whatever, when y'all go, then I can proceed. So the decomposition of organs, tissues, and muscles may appear to happen simultaneously, okay? But they don't, okay? There is a hierarchy of degradation. All right, and so that list that we kind of looked before, and here it is again, carbohydrates in the form of glycogen and glucose, they degrade first. Okay, that's the first thing to start to get broken down. Then the soft proteins begin degrading shortly thereafter. Those are the proteins that are present in the cells and in the tissues. So that's second to start to break down. Lipids are the last significant compound to degrade. And finally, the hard protein that make up the keratinized tissues, like hair and nails and so forth. And bone are the very last to degrade. So that's the hierarchy of degradation. So it may seem like, again, that
decomposition of the different tissues within the body system degrade simultaneously, but they do not. Carbohydrate decomposition. Because that's the first thing, right? That degrades. So carbohydrate. It begins with hydrolysis, right? Because you remember when we were talking about decomposition, we talked about that is basically the breaking down by water, right? And so the water content, you know, we talked about an abominous nightmare is an edematous body, because the more water content that you have in that body, it accelerates decomposition. So the decomposition of carbohydrates begins with hydrolysis, okay? The addition of water. We talked about that in chapter 18, of glycogen into glucose monomers. Okay, you see that? Glycogen into glucose monomers. So what is that an indication of? This is glycogen here, this molecule here. This is glycogen. With the addition of water, hydrolysis comes in, and it's been broken down into glucose monomers. Okay. So you remember when we talk about decomposition, we talked about the breaking down. When we looked at digestion, we looked at glycolysis. You remember when you're taking a large molecule, because that's what decomposition is. Large molecule taking that protein, you know, carbohydrate, whatever it is, a large molecule, and you're breaking it down into smaller molecules. Okay. Finally, at the end stage, this glucose monomer right here can be broken down further to final product of carbon dioxide and water. So as you can see there, that large molecule, hydrolysis, water splitting, breaking it down into smaller molecules, and it's being broken down further into final breakdown of carbon dioxide and water. Sugar decomposition in more detail. So in an oxygen-rich environment, aerobic conditions, we just kind of talked about aerobic and anaerobic conditions when we talked about, you remember those pyruvate molecules during the process of glycolysis. So fungi and bacteria, they decompose sugars such as glucose into glucuronic acid, citric acid, and oxalic acid. Okay? In an oxygen poor environment, that's anaerobic, without oxygen, bacteria decompose sugars into lactic acid, butyric acid, and acetic acid. Okay, and you remember in those two pyruvate molecules in that in glycolysis, when you're taking that sugar molecule that's being broken down, and it's broken down into two pyruvates, and in aerobic and anaerobic conditions where one goes on and is stored as lactic acid, and the other one goes on and attaches itself to that acetyl coenzyme A, which that is what goes into the Krebs cycle, right? And then from there into the electron transport chain in our ATP for our energy. So both degradation pathways contribute to the initial acidic environment associated with early stage decomposition. And then this picture here is just showing you. You have your glucose molecule here. Under aerobic conditions, this is the pathway that it would take. So you have glucuronic acid, citric acid, oxalic acid. Up under anaerobic conditions without oxygen, you'll have lactic acid, butyric acid, acetic acid. Okay, and we looked at this when we looked at the pyruvates. right? Oxygen levels dictate the types of acids formed from carbohydrate degradation. And so that was in glycolysis, that's what we started out with. We started out with that carbohydrate when we went through that chapter. This is just a reiteration of it. Protein degradation. Now just keep in mind a protein molecule, right? Protein molecule is a large molecule, right? Proteins do not degrade at a uniform rate because they make up such a diverse array of compounds. Proteolytic enzymes. They're involved in protein catabolism, right? They decompose first. They include such structures as the pancreas and the gastrointestinal tract. Over time, muscle protein, collagen, connective tissue, and reticulum fibers, they decompose secondly, after. The protein keratin, again, when we looked at the hierarchy of degradation, you remember that that was just before bone. So the protein keratin, which is found in the skin, hair, and nails, that decomposes last. Saprophytic bacteria are major cause of protein degradation. Saprophytic bacteria. What is saprophytic bacteria? It was one of your terms. We've talked about it. What does a saprophyte do? So saprophytes, they feast off of dead and decaying matter. Okay? Remember, we talked about the purpose of our embalming fluid. And we just talked about that a few slides back. And so the purpose of it, remember, is to kill bacteria. And then it's to, so it's a preservative and a disinfectant, okay? And so it does that. It kills the bacteria, and then it tries to pull together, cross-link those biomolecules who are starting to degrade, right? So when we use our arterial fluids, they're to kill the bacteria, and they're also to make conditions where bacteria doesn't have a food source. All right. So our cells also have an auto-destruct mechanism, which is both very cool and very surprising. So when the body turns acidic, the membrane surrounding the lysosome within a cell bursts. It's no longer caged by the membrane, and those lysosome enzymes begin to devour surrounding cellular structures. Putrefaction. So protein degradation process enzymatically driven with the aid of moisture, temperature, and bacteria content. Okay, so again, and which we'll look at hydrolysis, but putrefaction. Putrefaction encompasses 3 types of chemical reactions that lead to a myriad of interesting chemical products. So the putrefaction, hydrolysis. So as we see here, it's an amide bond breaking chemical reaction that involves the addition of water. That's what hydrolysis is. Okay, it's the splitting, the breaking down by water. Enzymes must be present to catalyze such reaction. This is favored in moist environments. Moisture, water. The ultimate products of hydrolysis are polypeptides, amino acids, and other amino acid links like oligomers. What do you see there? The ultimate products of hydrolysis are polypeptides, amino acids, oligomers. It's protein hydrolysis. What is decomposition? Bring it down.
Things that make it up.
Well, what is a protein? You remember we had a description of what a protein was? What is a protein? What does a protein consist of? You remember it was 50 or more? You remember the definition of a protein? It was 50 or more of amino acid links. You remember that? Less than that was a polypeptide. So it's showing you amino acids, you remember they're building blocks to proteins. So if I'm taking this huge molecule of protein, it's being broken down first to polypeptides, then from polypeptides to amino acid links and oligomers. Okay, so the progression is being broken down because you remember that we had the amino acids, we have the oligomers, we have the polypeptides, then we have the protein. So it's just going in the reverse, breaking it down. You're taking that large molecule, protein, broken down polypeptide, broken down further oligomers, broken down amino acids. Correct? So it's just showing you what is happening when you're breaking that large molecule down. Because remember, when we took a carbohydrate, we used that for an example. When we looked at the breaking down process, when we looked at glycolysis, and you remember, we took that large molecule and it was broken down to a monomer. Smaller molecule, okay? Smaller molecule in order for it to be absorbed into the intestine, into the bloodstream, okay? And for it to take the necessary pathway in order to be used in the end for energy extraction. So that's hydrolysis. We look still up under putrefaction, okay? Decarboxylation. What decarboxylation? Looking at the term, by looking at the term, what does that tell you that decarboxylation is?
Carboxyl or not?
What's that?
Getting rid of the carboxyl?
You got it. Decarboxylation is the removing of the functional group carboxylic acid. That's in the red. That's your carboxylic acid functional group. So this is the degradation process that chemically removes carboxylic acid groups. Decarboxylation, the removal of the carboxylic acid. So you're removing the carboxylic acid group from that amino acid and also from the terminal end of proteins and polypeptides. And that's what is shown here below. You're removing that carboxylic acid, okay? Which leaves you with a, what's this? What functional group is that? Did I hear? Did you say it?
Maybe carbon?
Well, I have a carbon attached to a. What two possibilities could this be? There were two functional groups that had a nitrogen. What were they? That's it, Bella. Amine. That's the amine. The other one is an amide. Remember that? Those are the two functional groups that had the nitrogen. So that's so decarboxylation. Again, so two common decarboxylation products associated with protein decomposition are putrescine and cadaverine. Remember earlier on we talked about when we looked at the functional groups and we looked at that amine, and we said that they were also referred to as putrescine, putrefaction. So when it goes through decarboxylation, and so what that is that if you take the a protein, arginine, and lysine, when you remove the carboxylic acid group from arginine, you end up with putrescine. Lysine, when you remove the carboxylic acid from it by way of decarboxylation, you end up with cadaverine. They both have, what function group is that again? Amine. Okay? They both have the amine functional groups on them, right? The only difference is the amount of carbons. Decarboxylation. You will need to know the difference between each one was decarboxylation. Hydrolysis, you should have that one down pat, right? And deamination. The deamination is the other. So it's also an enzymatically driven decomposition process that removes the amino group. Removes that amino group in the form of ammonia from amino acids and polypeptides. So decarboxylation removes the carboxylic acid. Deamination removes the amine group.
Yeah.
We've touched on the significance of ammonia formation as a protein degradation product in previous chapters. Who can tell me, who remembers this process here? What happens when this happens? The ammonia formation as a protein degradation product, what happens when we have that? Well, look right below it. Formaldehyde reacts readily with ammonia forming urotropin. You've seen that before, right? We've talked about this before. Ammonia reacts with formaldehyde producing urotropin, which is not helpful to us, right? So what happens here during the degradation process of the protein when we have this ammonia in the body? How is that a hindrance or a problem to us? Who remembers? Look at here, ammonia. The breakdown of the protein and it's producing all of this ammonia in the body. What's the problem for you as the ammonia? Well, that would be like a genesis, but here it's saying, because now when we have the degradation of protein, it releases a lot of nitrogen and ammonia in the body. So we have the ammonia and the nitrogenous waste. What problem does that create for you as the ammonia? And look at the problem there below. Ammonia plus the formaldehyde, which equals urotrophin. Real simple. Don't overthink it. Okay. I'm gonna need more formaldehyde. So you're letting me know I'm gonna need more formaldehyde. Why is that? I am gonna need more of a preservative demand. I am gonna need more formaldehyde. Why? You're right. I'll need more formaldehyde. Why is it?
More basic from all the nitrogen that I'm using.
What's that? You're on the right track.
There's more nitrogen from the urotrophin, so it's more basic. Creating a more basic.
And, okay, that's right, but what does that do? Why do I need more of formaldehyde in a case like this? What does all of this nitrogenous waste and this ammonia in the body, what does it do to my formaldehyde? That's right, it neutralizes. It neutralizes. Those of you who are at establishments, what is one of the things if you have a formaldehyde spill? What do? You neutralize it with ammonia. You throw some ammonia down, that neutralizes it. So I got all this ammonia in the body from the breakdown of the proteins. It weakens my formaldehyde, it neutralizes. So therefore, I'm going to need a higher demand of arterial fluid or solution, if you will, because the fluid is in the bottle, solution is what's in the tank. So I need a higher demand of my primary dilution factor. In a case like this, when we talked about advanced decomposition cases, we talked about those cases need a higher demand of fluid because in advanced cases of decomposition, you got a lot of nitrogenous waste and ammonia in the body. So I'm going to need more embalming solution, higher index, okay? More of a preservative demand. Additional decomposition reactions, sulfur-containing amino acids, cysteine and methionine. They're products of putrefaction as well. They undergo further bacterial decomposition to yield ammonia, foul-smelling thiols. You remember we talked about thiols. What is a thiol? What's a thiol? What do you see there in the red? In the red? What's this? In methionine. and then cysteine in the red. What is that? Sulfur. You remember when we talked about the functional groups and we talked about an ester, right? When you add sulfur to an ester, it becomes a thiol. That's when we talked about thiols, when we're going over the functional group, when we talked about esters. When you add sulfur to an ester, it becomes a thiol. Okay, so also pyruvic acid, sulfides, and hydrogen sulfide gas. They're known to have the unpleasant smells that are associated with decomposition. Okay, so you see there, you have the methionine and the cysteine, they're degraded. And so then you have your pyruvic acid, those that are listed, the thiols, ammonia, sulfides, hydrogen sulfides. Okay, is what you get from those two when they are degraded. So this is just a summary of protein decomposition, okay? You have your generic protein here. Hydrolysis takes place, okay? You have your polypeptides and miscellaneous oligomers here. It further gets hydrolyzed to a generic amino acid, right? Because look at the order it's going in, okay? Then we talked about deamination. Deamination was what? A removal of the amino group. So that's deamination, okay? Decarboxylation was what? That's right, the removal of carboxylic acid, okay? And so we have the arginine, and then we had our lysine. And so when we removed that, we ended up with both a putrescine and cadaverine, okay? And cysteine and methionine. Here are pyruvic acid. Then also we get our sulfides, ammonia, thiols, and hydrogen sulfides, okay? It's just the slides previously that we've gone over. This is just everything in a nutshell, okay? That we went over. Heme decomposition. Okay, remember when we talked about, we looked at this previously when we looked at hemoglobin. So we talked about it, and each color here is representative of the extensions of the protein chains, right? Each of them have an oxygen-containing porphyrin heme that's nestled within it, and that's the little pink structure with the red circle in it. That's for each one of those chains, if you will. Now we've seen how proteins can decompose. But also of interest to us is that the decomposition of heme itself. So during normal catabolism, you remember when we looked at metabolism, metabolism deals with catabolism and anabolism working together. And catabolism is what? That's right. Broken down. Catabolism is to break down. All right. So during normal catabolism, heme gets broken down into green bilirubin and eventually to yellow bilirubin. Bilirubin is eventually secreted as bile from the liver. And so here we see the process of the breakdown of heme to the bilirubin, which is green, and further reduced into the bilirubin, which is yellow. Okay. In cases where we have that yellowing pigmentation, you get that seepage into the capillaries and into the surrounding tissues. Okay? So in cases where you have that yellowing, that jaundice associated with some type of liver disease. And that results in the bilirubin coloring that we can see in some of the cases that we would get. And that's due to the buildup of the yellowing color in the tissues, and that's due to the obstruction of the biliary tract in the liver. Okay? And so we get that yellowing color. Now here, and some of you, hopefully you haven't experienced it, you did better. But if you're not careful, during embalming, the yellow color due to the jaundice can be converted into an even worse green color. Okay? So we have lots of different types of chemicals that can be used to aid us in cases like this, right? Some of you are maybe familiar with some of the John Dyer fluids, right? That are supplementals that you can add to your cocktail, if you will, and to aid in these cases, right? Because lots of cases may have different factors, right? You may have a case that may be edematous, you know, that you need a higher index, right? But if it has this jaundice color, you have to be careful with that, because the higher the index, you run the risk of that body turning green. Okay, so you have to be careful in adding whatever supplemental chemicals that are needed in cases like this and even some others. But also you would use help with some type of makeup when you're cosmetizing, some opaques. You would have to use in cases like this. So all of those things, the different types of supplemental chemicals that we have along with our cosmetizing. Nucleic acids, they decompose into sugars, nitrogenous bases, and phosphates, okay? Because you remember we talked about nucleic acids, what are they made up of? Well, look at it. Look at the structure here. This is a nucleic acid here. This is one here. This is one here, and this is one here. Remember when we talked about the nucleic acids? They have how many components to them? How many components do nucleic acids have? How many parts? How many parts? Who said that? Would you know? It's 3. Well, right here. Each nucleic acid has a sugar, has a nitrogenous base, and has a phosphate group. Okay? The sugar, the phosphate group, and the nitrogenous base. That's what makes up, those 3 components make up a nucleic acid. Now, we just talked about the breaking down of sugar. We talked about that when we looked at it in metabolism. We looked at it. But the nitrogenous bases, they break down further and release ammonia. Phosphoric acid is released into the body as phosphates. So we look at the different nucleic acids, this nucleic acid broken down into our phosphate, our sugar, and our nitrogenous base. So it's taking that nucleic acid is made up of three components, breaking each one of them apart. Okay. Same thing with lipid decomposition. Lipids. So adipose tissue or body fat make up a large portion of the lipids that's in the human body. All right. Triglycerides are the most common. Diglyceride, phospholipids, and sterols are also quite prevalent in the body. And so when we talked about lipids before, we looked at all of those classifications that fall up under lipids, okay? So phospholipids, remember they were a major part of the cell membrane, right? And why were they a major part of the cell membrane? Remember when we looked at the cell membrane, it was a round structure. Took a piece of it, and then it has the hydro, and then the tail. Okay, so that's why they're used for as part of the cell membranes. So phospholipid decomposition. They decompose to phosphates, triglycerol, and fatty acids. We're taking this phospholipid here. Okay, and so all the components that make up this phospholipid, when you decompose, this portion here is your triglycerol. These here are your fatty acid chains and your phosphate. So it's just taking it and splitting it down. Okay, breaking it apart. But Max, I thought you had a question. You have a question?
No.
Sterols, we talked about sterols. They were class under lipids as well. We went through lipids before sterols. Remember, this is the structure of our sterol. Okay, had the 3 6-member cyclic structures and the 5-1-5 cyclic structure. So the derivatives are oxidized into a plethora of compounds, which we see here. Okay, the aldehyde, the ketones, and other miscellaneous organic compounds. All these here are ester. Talked about ester, the alcohol, ether. And so, but the final decomposition, it gets decomposed further into carbon dioxide and water. Triglyceride decomposition. Again, same thing. Our triglyceride, that decomposes. It's the breaking down by way of hydrolysis. And so it's that breaking down of the fatty acids to the fatty acids and the glycerol backbone. Okay, so this is our glycerol backbone. These are our fatty acid chains. As you can see, the whole structure here is taking the glycerol backbone, breaking that apart from each one of these fatty acid chains. So once the fatty acids are formed, they can be, so on previous slide, these fatty acid chains from the glycerol backbone is broken apart. So now we have each one of these fatty acid chains, right? And so they can be further decomposed depending upon the aerobic conditions or anaerobic conditions, okay? And so up under the aerobic conditions, it can be further decomposed into keratin, aldehydes, further decomposed into carbon dioxide and water. Again, Once the fatty acids are formed, they further degrade in an aerobic pathway or an anaerobic pathway. Now, both of these pathways will happen simultaneously. But again, finally decomposed into carbon dioxide and water. You'll see that as a question. One of the questions will be, what's the final degradation of a triglyceride or a lipid, carbon dioxide and water? Grade wax. So under anaerobic conditions, that fatty acid can be transformed into grade wax, or also known as adipocere. Fatty acids produced during decomposition that get saponified. That was one of your glossary terms, saponification. And that's a soapy-like substance, so a wax-like soap. That's what saponified or saponification is. Keratinized tissue and bone. These are the last things. We're going into the order of the hierarchy of decomposition. So now we get to the last thing that decomposes, and that's the keratinized tissue and then bone. So the hard proteins that make up keratinized tissues, such as the hair and the nails, are among the last compounds to degrade. Okay? The very last thing is bone. So proteins that we've looked at, they're broken down into polypeptides and other oligomers. They're eventually reduced to the constituent amino acids. We kind of talked about that, right? So those proteins, they can be broken down first to polypeptides, then from polypeptides to oligomers, and then from oligomers to amino acids. Amino acids are building blocks to protein, okay? And so eventually the last thing to decompose is bone, okay? Eventually the rest of the bone will disintegrate into dust, into the wind, which is where we come from. So that's the last thing to decompose. All right, so going through the hierarchy of decomposition, the order in which things decompose may seem like that they all do it simultaneously, but they do not. Okay, and so know the order in which decomposition takes place, and you may see a question asking about the order in which a protein. So we go from the large molecule to a polypeptide, from a polypeptide to an oligomer, from that to an amino acid. Okay, take 10 minutes, we'll come back and we'll hit chapter 24.